This is another episode of Dr. Lanny Health. This one in particular will deal with the discovery of protocitric acid. You heard me say before, there's nothing new under the sun. However, there's much yet to be discovered here. This podcast and others like it are devoted to the fundamentals of the art and science of discovery. And I'm going to deal today with the fundamentals of discovery as we go along and discuss how I came about to learn about PCA. First of all, every discovery has to be based on the fact that there must be a need. The patent application, for instance, on any kind of invention says you must meet the criteria of a long-felt unmet need. And what would be the long-felt unmet need in this particular application of protocitric acid? It's arthritis. So common: osteoarthritis, psoriatic arthritis, rheumatoid arthritis. And all of the present drugs treat or control only the inflammation. So any drug that treats arthritis causes the joint lining to produce a substance that reverses the inflammation within the fluid of the joint. None of the present osteoarthritic drugs affect or help or improve or heal the cartilage. Protocitric acid does that. So the first thing you have to do in looking at invention or discovery, you've got to have a curiosity and you've got to make observations. And you must continue to be curious and continue to be observant. I mentioned before, I'm Lanny Johnson. I'm a medical doctor. I'm a holder of 100 U.S. patents. Every one of them was a fabulous idea, but only a few of them ever materialized anything. In business, we like to qualify a customer, and I may be qualifying you when I tell you what the outcome could be of having an invention or an idea you want to be an invention. Discovery takes a lot of time, a lot of energy, and somebody's money to translate to success. And here are the facts regarding patent applications. 40% of patent applications may be approved, and of those approved, only 1% ever have any commercial value. So most people that hold a patent, the reward that they get is a commemorative plaque on their wall at home and no compensation other than that. Unfortunately, I have beat the statistics in the past as an orthopedic surgeon. I was one of the pioneers in arthroscopic surgery, and I developed the motorized instruments for arthroscopic surgery that made that possible. And as a result, I was rewarded. But in this particular area, I'm outside my expertise in this venture. I'm not a biochemist, I'm not a bacteriologist, and I'm not a nutritionist. So my question is, am I going to catch lightning in a bottle twice? I don't know. We'll find out. The principles, though, remain the same. Here's my story about protocitric acid because it's hard to pronounce correctly. I've said that before. It's spelled in a way that it doesn't tip you off to how to pronounce it, so I'm going to use the acronym. As you can see, PCA comes from the letters that are within that wording. Until recently, I never heard the word protocitric acid. I doubt if you have either. My medical training was all influenced by big pharma and pharmaceuticals, and there was never a thought about nutraceuticals. Protocitric acid, or PCA, happens to be a nutraceutical. It's a food substance. I mentioned before that it's in actually our human diet. It's in throughout nature, in the ponds, in the trees, in the soil, and it also, the microbiome in our large bowel makes a small amount of PCA every day, so it's completely natural. My focus in the past was always on anatomy, pathology, and or surgical technique, and as such, I had a real interest in cartilage healing, and I published on that over the years. So my first time that I was alerted to get involved in this was in 2005. I saw an article in the Lansing State Journal, a newspaper in Lansing, Michigan, that some scientists at Michigan State had discovered that if you put the red dye of cherries on the islet cells of rodents, the pancreas, it would make insulin, or more insulin. I then reasoned, well, maybe it's possible that if these same dyes are put on human joint lining in a laboratory test taken from humans during surgery, with their permission, of course, and we found out that the application of the dye, which is called cyanidin 3-glucoside, applied to human synovium topically, increased the amount of IGF-1, or the human growth hormone. So now that perked my curiosity, but I then made to remind me, why am I so curious? Well, here's the thing. As an orthopedic surgeon, I had interest in cartilage healing and published on that. And I knew that the growth hormone IGF-1 for the last 50 years was known not in humans, but to promote articular cartilage healing in various applications. Then about 30 years ago, I was a consultant with a company, Chiron, in California, where they gave recombinant IGF-1 by injection, and we had before and after findings, and we saw that the articular cartilage lesions, heretofore not known possible, were healed. So we knew more evidence that now in a human, IGF-1 would make a difference. So then imagination is so important. You have to think forward as what's possible. And I imagined the potential that there might be a solution. And so as a result of the tests that we did at an independent contract lab, that we learned that cyanidin-3-glucoside was rapidly changed to protocotric acid, or PCA. And so it was unlikely that the parent cyanidin-3-glucoside, the red dye, was the issue or the active reagent. It was probably PCA. So then what we did was we said we need to go beyond the human test. We received 2 patents on the use of cyanidin-3-glucoside for treatment of arthritis and stimulation of IGF-1, or the growth hormone. But it was rather impractical since we already now knew that it was the metabolite, PCA, that was the active reagent. We then pursued this further with some studies on animals, where we gave one group of animals protocotric acid by mouth and the other one by injection. And what we found out was that by either way, the PCA resulted in reversing the arthritic change in the animal. And so what was interesting was that we not only reversed the inflammation like every other drug did, but the PCA taken by mouth, for instance, resulted in the joint lining producing IGF-1, reversing the arthritis in the synovium and the synovial fluid, or the joint fluid. But rather importantly, and unlike any other, it affected the integrity of the cartilage and regenerated in the cartilage. And so we had evidence that the lubricant, which is on the gliding surface of cartilage that makes the cartilage move easily, that was increased. And the structure of the articular cartilage is based upon 2 products. One is called aggrecan and the other is type 2 collagen, and both of those were increased. So we found out, excitingly, that the PCA by mouth, or actually by injection as well, would reverse arthritis and also repair the cartilage. With that excitement, we then applied for a U.S. patent, and that was about in the year 2010. And what happened was it got rejected. And even though we showed all the differences that I just mentioned, this patent languished and was refused for 14 years. Well, anybody involved in discovery better have some resiliency, because you're going to meet disappointments along the way. I was really upset, so I went to Washington with my lawyer and met with the patent attorney and made an impersonal plea, only to be rejected. Still no patent. So I would recommend that if you're in any kind of discovery, you better have a plan B. I was discouraged. I wondered, okay, what am I going to do? I'm sitting here, I can't get this. I went through 3 different patent attorneys. I went through 3 different examiners over the period of 14 years. But during that time, I wondered what else protocotric acid would do. And so I started looking in the literature while I was waiting on this primary interest. And what I found out was that in the literature, as I may have told you before, PCA was first in the literature in 1890. It also had 1,000 publications in the literature. Wow, I didn't know that PCA had so many potential applications. And so I remained persistent, and I started looking at what other things this could do. And during this delay, over the 14 years, I have now 31 patents on PCA, which was a positive response to the rejection of what happened originally. And now I have a greater number of intellectual properties. And so now what I'd like to do is I'd like to review the patents that were issued over time. We'll tell you the many applications that there can be for PCA.